Hello, everyone. Welcome to DNA Dialogue, where members of the genomics community in Australia and beyond can hear about new developments in health genomics internationally. I'm Tiff Bortwood. I'm the Managing Director of Australian Genomics. As we come together virtually today, we acknowledge the traditional custodians of country throughout Australia and their connections to land, sea and community. I'm joining this webinar from Naarm, the lands of the Wurundjeri Woiwurrung people of the Kulin Nation. I pay my respects to Indigenous elders past and present and extend that respect to all First Nation peoples today. Please ask any questions you have during the presentation today using the Q&A function, and select questions will be read out and addressed by our speaker during question time at the end of the talk. If you're active on social media, please hashtag DNA Dialogue and tag @OzGenomics. So it's my pleasure today to be joined by our speaker, Dr. Ana Topf. Ana is a Senior Research Associate at the John Walton Muscular Dystrophy Research Centre in the UK, where she coordinates the next generation sequencing projects. She's a molecular biologist with over 15 years' experience in the analysis and interpretation of omics data for rare disease, particularly neuromuscular and neurodevelopmental conditions. Throughout her career in NMD, Ana has co-authored over 100 publications, including the discovery of new disease genes and pathomechanisms, and novel digenic model for congenital skeletal myopathy. Genomics has revolutionised diagnostic process for patients with neuromuscular diseases. However, access to sequencing technologies in less developed regions, such as Latin America, is not only limited, but it's extremely variable. So today, Ana will be sharing us with the story of Latin-SEQ, which is a consortium of neuromuscular units from Latin American countries that are focused on diagnostics, basic research and training in the neuromuscular dystrophy field. Ana, thank you so much for joining me.
Thank you very much, Tiff.
Welcome. Thank you for joining us at this early hour for you. We're very much looking forward to hearing your presentation. That looks great. Thank you.
Excellent. Good start. So thank you very much for having me. As Tiff said, I work in the John Walton Muscular Dystrophy Research Centre, and I wanted to tell you a bit about our new baby in our sequencing projects called Latin-SEQ. And this is looking at diagnosis in Latin American countries, as introduced. Yes. So the background of this project, I would say, that is based on our large experience that we have at the John Walton Muscular Dystrophy Research Centre in MGS projects in general. So our first large project started about 10 years ago, called MyoSeq. And in MyoSeq, what we did was we collected a very large population of patients with neuromuscular conditions all throughout Europe and the Middle East, and tried to provide a genetic diagnosis for them. We applied exome sequencing and looked at singular nucleotide variants, but also copy number variants. We had a very successful diagnostic rate of about 55%. But it wasn't just diagnosis. We discovered new genes, we established new disease associations, phenotype, genotype correlations, and so forth. But it wasn't just the output. It was what was very interesting about the collaboration was the enormous input that we had from all our sites and how prolific the project was, even from the publication point of view. So we are very happy with the output. It's a project that keeps giving in a way, because even though we have a very large or important diagnostic rate, we still have an important number of patients that remain undiagnosed, and these are still being reviewed. But we learned a huge amount of how to process the data, how to collaborate with the participating centers and how to create an environment that is rewarding for all the sites. Now, something that happened during this during MyoSeq is that you probably might have realized by now that I'm not from the UK. I've been in the UK for a very long time, but I'm not from there. So something that I thought while working in this project is that these patients would have had access to genetic testing at some point. So most of the countries that are involved in or were involved in MyoSeq were countries from Western Europe, with some countries in the East and also Middle East that maybe had less access to genetic testing, but I would say that the vast majority did. And this is not, as Stephanie was saying, this is not the case in other regions in the world, and in particular in Latin America, where I'm from. I'm originally from Argentina, came to the UK to study when I was younger, did my PhD and decided to stay. And this was setting up this project was in a way trying to give something back of what I learned while I was there, and trying to continue with my connection and my roots with the countries. These are regions that are obviously understudied. There are very few publications looking at genetic prevalences of different diseases or neuromuscular diseases. And as we were saying a couple of times, there are unmet needs. What's very interesting is that despite not having access to genetic testing, most of the clinicians trained in Latin American countries have a vast and very thorough clinical expertise. So what they lack in genetic testing, they have in clinical experience, and most of the diagnoses are done this way. So that's how Latin-SEQ was created. We say that Latin-SEQ is the younger sister of MyoSeq. So the aims of the project obviously are to provide genetic diagnosis to these patients, neuromuscular disease patients in Latin America. We want to learn about the prevalence of the neuromuscular diseases in the area. As I said, there's not much done about this. We imagine that we will identify variants or even genes are specific for the region, in particular genetic variants that might be founder variants. So we want to look at these in this as there might be specific genotype-phenotype correlations. But the other aspect of the project that we want to develop is to try to support local development and help the health providers and the health members to participate and to grow in this project as much as possible. We didn't want this to be just us providing a service, but trying to make or to become a community where we could work together. So with all this in mind, we wrote up a proposal and we tried to get funding. We were lucky enough to make some pharmaceuticals companies interested. So currently we have funding from Sanofi, Roche, and the Jane Foundation, as well as a small patient organization, Cure LGMD2I. We managed to get funding for exome sequencing for 1,000 patients, and this in theory should last for 2 years, although we hope that we'll get extensions and we can continue this because we see this as a very large project that should hopefully could continue in the future. We started in May 2023, so we've been working on this for already a year, and setting up the consortium and the initial stages obviously proved more difficult than we expected, and I'll mention this in a minute. So the outcomes of the project are obviously first to provide this genetic diagnosis to patients that would not have access to this. The basic research is, of course, another interest of us and the clinicians involved. So again, anything to do with new phenotype, genotype associations, new gene discovery is something that we know that the project will lead to. But the other arm of the project is something that I mentioned, this virtual network that we wanted to create. So we wanted a virtual space for clinical and genetic development in the neuromuscular field for everyone that is participating in the study. So we wanted the centers, as I said, to become part of the diagnostic process and to work with us in this project. So what we are doing for this is that we're planning head-to-head meetings, and this will be direct meetings from the Newcastle team with each local site, where we will first feedback the solved cases, but then discuss anything that is any case that remains unsolved, looking for additional clinical information or further tests that could be done. And then what are the following options if we want to do another type of analysis, either a genome or other type of genetic studies. And this is what we want to be very collaborative and a discussion, a two-way discussion for each case. We also we're planning virtual webinars or workshops, and this will be every 2 months. We started already. This will be done either by our local team, but also our Newcastle team, but also the local participating centers or other international speakers. And we thought of different training topics or relevant topics in the field, either the NGS diagnosis of neuromuscular disease conditions, or how to use MRI tools for diagnostic purposes, themes associated with genetic counseling, or how to follow up patients. Again, we are open to discussions and anything that the consortium thinks that is relevant for the webinars will be taken on board. So for the recruitment of the patients, what we decided was that we wanted to include as many patients as possible. This is different to what we did in MyoSeq. In MyoSeq, we concentrated on patients with limb-girdle muscular weakness, so they within not an early onset, so adult onset. Here we are not restricted by that. We wanted to include conditions that could be caused by a nerve, a neuromuscular junction, or a muscle issue, but that they had a suspicion to be genetic. So we want the clinicians to, as much as possible, try to rule out anything that could be an acquired condition, because obviously we wouldn't be able to diagnose these. There's no age or sex restriction for the inclusion of patients, and the only clinical exclusion that we have are the myotonic dystrophy 1 and 2, and this is FSHD, and this is mainly because we wouldn't be able to diagnose them easily with exome sequencing. And we are giving priority, if possible, families that have multiple affected members and those from ethnic minorities. The way that we designed the study, during recruitment, we of course recruit the patient or the index case, but we also want to have relatives, both affected and unaffected, from the very beginning of the project. And this again is from previous experience, where we know that any additional DNA or biomaterial that we want to collect afterwards becomes much more difficult. So we've asked clinicians to, once they call the patient to clinic, they get the whole family or as many family members as possible, and this will help enormously in the analysis later on. The strategy for the analysis, we want to do first a single gene pre-screening, and this is based on the clinical suspicion. And after that, those that remain negative will be exome sequence and a proband only base. So as I was saying, we're not sequencing family members initially, but only the proband. And if needed in following stages, we will do that. The data analysis, we're doing an in silico neuromuscular disease panel of over 600 neuromuscular disease genes. And in cases where this proves negative, we will do a phenotype-driven analysis, looking outside those neuromuscular disease genes. So for the phenotype information, we try to have what we call deep phenotyping. and we're using GPAP, it's a genome phenome analysis platform that was developed by RD Connect. That this is an infrastructure, a European infrastructure for rare diseases that we've been collaborating with them for a long time. And it's basically a clinical database to archive standardized clinical data. So here the clinicians, the participating centers, will be able to upload any clinical information for their patients, onset, progression, inheritance, all the presenting symptoms using HBO terms, as well as any lab results, images from MRI or muscle biopsies, and images from clinical notes and pedigrees as well, where we can identify all the affected family members and any other information all we want. So this will allow us not only to compare the clinical information for each patient, but in the future, if we want to do any cohort analysis by specific phenotype, for example, we would be able to do that. So the workflow for the project, obviously the patient selection, the sample recruitment, and the phenotyping is done by the participating centers in Latin America. The samples are sent to us in Newcastle, where we do the sample processing, DNA extraction, QC, and single gene pre-screening if needed. Samples are then sent to our sequencing provider in Barcelona, where we are working with the National Center for Genomic Analysis. The data is then uploaded onto a genomic platform, again developed by RV Connect, where in principle the team in Newcastle are doing the data analysis. But this is again something that we would like the centers to be involved in the future. And then we have these one-to-one case discussions with the clinicians that will inevitably result in a feedback and a second look and second round of data analysis that is usually even more productive than the first round, where we manage to discuss almost individual cases one by one. And these are done in these sessions that I mentioned before. So as I said, the project started over a year ago, and the initial stages were quite difficult. Bureaucracy is high in Central America, in South America, and in our countries. So we knew that this would be difficult, but we are very happy to say that now we managed to enroll over 60 sites in the project. These are from 32 cities in 18 countries in Central and South America. And there are different stages, of course. For each of these centers, we have to sign material transfer agreements. The centers have to pass the proposals through the ethics committee, being local or sometimes in the national committees, and there are consents to be approved. So local consents as well as our biobank consent. So I have to admit that not all the 60 sites are totally up to date with this. They're in different stages of the paperwork, this part of the process, but they are all well on board, and we hope that all of these 60 sites remain in the project. For the collection of samples, again, we knew that this was not going to be easy. Of course, not all the centers, these are hospitals or small referral sites, do not have access to extraction of DNA from biological materials. So even though we would prefer to have DNA as the material sent to us, we know that that's not the case. So we accept whole bloods, salivas as well, and now we recently started collecting dry blood spots because some of the centers do not have access to fridges or freezers to even store the bloods until the shipment. So we're trying to maximize the way to be able to collect and recruit patients. Also because sometimes patients do not come to clinics as often as they would like. So having access to saliva collection, for example, makes the collection of material slightly easier. So this is the breakdown of the samples we've received so far. So only a third of the samples received are DNA, the other ones are saliva, fresh bloods, and a few dry blood spots that we are currently extracting. Sample submission, we knew that this was going to be, because it's a large project, we knew that it would be difficult. We had issues in the past with this. We tried to maximize everything that all the mistakes that we had in previous projects, we're trying to get to avoid those as much as possible. So every single patient gets given an identifier. Each center has their own identifier, and each family as well. There's cross-reference of all the samples and families to make sure that there's no issues with this. We collect information about relationship status, whether the clinical status, of course, sex and age. And something important is the suspected clinical diagnosis. So this is not a genetic diagnosis, of course, it's what the clinician at the time thought that was the differential diagnosis for that patient. And we also collect information about the previous analysis, if any. So some of we were saying the access to genetic testing is very variable in Latin America. So some centers do have access to some minimal tests, but not all. So this again help us to decide exactly what type of test we will do in each case. So we're doing a very much personalized type of approach. We thought of we wanted to be as patient-centered as possible. This might sound totally trivial, but we again international shipment from South American and Central American countries is not easy, not even for us to send material to them. So again, we try to facilitate this as much as possible and to minimize issues. We've got SOPs, videos. We tell them how to pack all the material. We've established, we've got a corporate account with one of these carriers that help us to make this movement as swift as possible. Again, we wouldn't expect to have these issues, and we didn't have these issues when we were working with European countries, but this is not the same. So again, it's something that is not trivial, unfortunately. So finally, by the end of last year, we started to receive samples. We already have a total of nearly 488 biological samples, almost 50-50, just slightly more males than females. And you can see here the breakdown by age. So as I said, we include both pediatric and adult patients. And here's the breakdown depending on what type of test were done. So as I said, the vast majority of nearly 75%, or nearly 3/4 of the population do not have access to any genetic testing, with some having access to maybe small panels of 10 genes, panels of neuromuscular diseases. This could be up to between 50 and 100 genes sometimes in a few cases, and just a 6% access to exome sequencing. So the reason why we still included patients with a negative exome was because, as I said, we still want to try, if these are still negative, we still want to try to find a genetic diagnosis for these cases. And what we hope is that maybe we will, for these patients, we will not do the West as initially thought of, but discussing the cases in more detail, it might be that a different type of approach is necessary. So obviously the project is not high tech initially, is just try to provide this much needed genetic diagnosis, but at the same time, we know that if we've got cases with negative exomes, we are happy to, in some particular cases, go for long read sequencing and see if we can identify something new that has been missed initially. The other aspect that we wanted to carry out in the future is to have access to the raw data of patients that have been, of the few patients that have been previously analyzed, because we know that most of these tests were done by private companies that obviously do not look at the genetic data. Probably shouldn't say this, but from the same, with the same interest that we see. So we found that the companies that, in some cases, companies that did Some of the genetic testing were not reporting single hits, for example, and other issues. So we would like to have access to this data, raw data, to be able to reanalyze and see if we could gain something more. The breakdown by site. So here you can see all the countries that already sent us material. Not everyone managed to collect biomaterial from family members, but most of them did. And the breakdown by disease group, again, this is the clinical suspicion at the time of recruitment. So the most common suspicion is a general myopathy, followed by neuropathies. We were not expecting this initially, but also followed by other muscle conditions. We imagine that these are more complex conditions that were not described by our initial classification. So based on this clinical suspicion is that we guide the single gene pre-screening, as I said. So those patients with a suspicion of Duchenne or Becker muscular dystrophy, those with suspicion of spinal muscular atrophy or neuropathies caused by PMP22 copy number variants are tested initially by MLPA. And those patients with a clinical suspicion of congenital myasthenic syndrome or mitochondrial myopathies are tested by point mutations. So we know that when these are recurrent mutations, that we know that solve a good proportion of cases when tested initially. So all of the ones that remain negative move on to the second stage of the exome sequencing. So of these, I know that there are still small numbers. We tested 37 patients and we already have some diagnosis accounting for 35% of the tested cases, which is more or less in line with what we were expecting for this type of analysis. So those that are negative for the single gene screening or that didn't have any pre-screening go through exome sequencing. As I said, we only just in the last 2 months we've received the first batch of sequences, and last week the very last one. We have 43 exome data sets. As I said, we do a first in silico neuromuscular disease panel for over 600 genes. And we are first looking at single nucleotide variants and small indels. At a later stage, we hope to look at copy number variants as well, of course. And here we've got over 50% of diagnosis or expected diagnosis. We've got cases that are solved, clearly solved. We're doing all the variant interpretation following ACMG guidelines. The likely solved cases are cases where we found pathogenic changes, but maybe we still need to do some confirmatory analysis as segregations to determine the phase of those changes. And we've got a small number of single hits where we identified pathogenic changes in recessive genes, and we're still looking for the second change. But we know that these are very likely to be disease causing based on the phenotype match for the gene in question. So here is the breakdown of the genes that we have identified so far. This is slightly unexpected in a way. We've got more than, so we've got 50-50 more or less, so 50% of genes causing neuropathy and the other 50 are more muscle related. But I guess that this is in line with the clinical suspicions that we had. So just to summarize, looking at the three aspects that I mentioned about the project, I think we are fully on board with the genetic diagnostic side. We've got both for the single gene screening and also the WES, where we think that the levels of diagnosis are what we thought we would have, or maybe even more. For the virtual network, we started one-to-one meetings with the two centers for which we already have exome testing, Uruguay and Peru. We started the monthly webinars. We've done 3 of these already. We've got a website we'll have, this is again a lot of work, unfortunately, where we'll have information about the whole project, all the centers, and all the developments. And from the basic research, although of course, we imagine that from the exome data, we'll have case reports and many other things that will follow up as the case that I just mentioned. But in parallel to our Latin-SEQ study, we've got already two basic research studies. One is the NAV study, where we're looking at expression of dysferlin in neutrophils from patients with neuromuscular conditions, and try to see if we can use the expression of dysferlin in this material as a diagnostic tool to help diagnosis. And the other one is the Latin-SEQ Plus, where we look at the impact of genetic testing in patients and families, but from a genetic counseling point of view. So this is a summary of our project. We feel that now the project is at the starting to, it took a very long time to get the cohort established and the centers recruited, but we are now finally moving forward and starting to get all the interesting results and providing feedback to the centers and having these discussions that we feel are incredibly rewarding for all of us. So I want to acknowledge, of course, all the participating centers, and in addition, Alejandro González Chamorro, that's our project coordinator. It would be absolutely impossible to have this without him. My co-PI, Jordi Diaz-Panera, the NGS team and the biobank team, and our colleagues in Barcelona who are coordinating the webinars, and of course our funders. And this is a picture of us in the last EVELAM meeting last year in Bogota, where we first met some of our collaborators. We hope that in the next meeting we'll be able to have a picture with all our clinical colleagues participating in the study. And if you've got any questions, I'm happy to take them now. Thank you very much for listening.
Ana, thank you very much for that walkthrough of the Latin-SEQ project. What an extraordinary amount of, I can just imagine, in fact, I'm just getting slightly activated, the idea of the logistics involved in establishing all those material transfer agreements, etc., let alone the challenges of the operational logistics in the model of having sort of the sample processing in Newcastle, and then the sequencing in Barcelona, and then the data analysis. What are the issues? I mean, we won't even touch on the establishment, but congratulations on setting up that infrastructure for the program across 18 countries. In the terms of, I mean, the logistical challenges I understand with the sample transfer, are there any ethical concerns of participants or families associated with the samples leaving, their countries and being processed in Newcastle?
Yeah, so this doesn't come from the participants themselves, or nor comes from the clinicians. When we had issues like that, it came more from governments or local regulations about samples leaving the country. We don't have major issues. In a few countries, in addition to the ethic approvals, then the participating centers need to get an approval from the government, from the law, or from some government, some political institution that gives permission to take samples out of the country. We haven't had many of these. I think it's only 2 cases where we have this issue. But this doesn't come from patients themselves, I have to say.
Okay. And also, just to your case, I did have a peek at attendees, and there are some clinical geneticists online that may have some thoughts. So if you do, and I'm not going to name and shame, but if you do, please pop those in the chat and I can relay those to, or in the Q&A rather, and we can discuss those as well. Another part I wanted to ask you about in a little more detail is the industry funding, and whether there are sensitivities associated with sort of public-private partnerships with the project, concerns about potential access to data? Like, is their involvement purely philanthropic, or is there sort of access to outputs or data or anything else from the project?
No, they don't. And this was clearly set up when we signed the contracts with the companies. The access to the data, the only access to the data they have is aggregated data. We don't share any, of course, any personal data. I mean, I didn't even mention this, but all the data is pseudonymized. There's no information about, there's no personal data shared with us directly. It's just the clinical data. But no, the funders do not have access to specific or granular genetic or clinical data. They will have just general outputs about diagnostic yield and genes identified, but no more than that. And there was I think there was a second part to your question.
Oh no, I think you covered it. That's
No.
fine. We have a question from Radistina as well. Hi, Ana. It's fantastic you're doing this project to give back to your country of origin and other countries. Firstly, interested in how did you start the project? Who did you contact first? I imagine strong networking connections are essential. And then we might follow up with her second question once you cover that one.
Yeah, definitely. Again, didn't mention this, but when our colleagues in Barcelona that are part of the coordination of the team, of the project, sorry. Again, these are colleagues that we've been working on for a long time. One of them is from Venezuela, the other one is from Honduras, and they've been working with, we've got already some colleagues over there. Most of the work that we do is just, through personal communication, let's say. So the very initial stages, what we did was to try to establish a list. Well, we got a list of neuromuscular clinicians working in Latin America. We got in touch with them, asking initially if they had any patients that were not diagnosed, if they were happy to collaborate or participate in a project like this, and which infrastructure they would have or they would lack. And with that long list of patients that was our preliminary data to go back to the funders and see if this there would be interest. But it was just personal contacts, really. It's just years. I'm not a clinician myself, but all the clinical, most of this, as I mentioned, the clinical training in these countries is incredibly good, I have to say. So the clinical experience is amazing. Maybe because they lack other aspects, so all the diagnosis is done clinically. Those countries or those centers that have a little bit more access sometimes do train in other centers in Europe or not so much in the States, but mainly in Europe. And that some of the connections were done that way. So the team in Barcelona, our colleagues in Barcelona and ourselves had just previous connections. It's just experience in the field and networking, previous networking.
I was very interested, you made a specific point about the excellence of the clinical training and the strength of the clinical suspicion or diagnosis that's received, which, in terms of your results and, the fact that, 35% are resolved in the pre-screening process just is a testament to that, isn't it?
Yeah, absolutely. Yeah. And again, this is half and half, their clinical experience and our genetic experience, knowing that because previously we had a 20% of solve rate when we did single gene pre-screening. So this is even better. And of course, we're avoiding doing an exome that it won't be necessary. So we are saving money that we can use in other ways. So
yeah, absolutely. So another question around community engagement. And did you have any of the patients or family members involved in any stages of the project as collaborators in the research activities?
Not so far. Not in our project at the moment. our parallel project, or this project called Latin-SEQ Plus, that looks at genetic counseling and the impact that having a diagnosis has both on family, on patient and family members. There we will try with the PI for the project is a completely separate project that is using our data and our results to feed into this. The genetic counselor that is working on that will try to have access to the patients and have direct discussions with them for this. So far, we haven't had anything. I have to say that it's not so easy. So access to patients. So the patients are seen in clinic regularly sometimes, but not as often as they should be probably. And this is related to commuting issues and other just financial problems in the region, of course. So we don't know exactly how easy it will be to, because funnily enough, when this project about genetic counseling was developed or was thought of, the lead thought that they could try to establish some type of survey that could be done online, so the patients would have access to an online survey, but that only shows how we see things sometimes from a completely different perspective, because access to an online survey, I don't know how many of those patients will really have access to that. It's not that they won't have access to telephones at all. I don't want to paint a completely dark image, but it won't be as easy as we would think. But yeah, hopefully at that stage, we'll have direct participation of the patients. Yeah, but not so far.
Okay, and that actually preempts Erin's question, who was sort of saying, what has been the involvement, but also she queries, the impact of a diagnosis for families. And that was a question I wanted to tease out with you myself a little bit, because across 18 countries and, 60 different clinical sites, there'd be a whole spectrum of available sort of levels of maturity in downstream interventions after achieving a diagnosis. So I think this, Latin-SEQ Plus project going into the significance of a diagnosis for families, and reproductive confidence restoration and all these different outcomes for families will be super interesting.
Yeah, and also something to remember that some of these countries are probably more religious than what we are used to in Europe, and there are other cultural norms and many different social and cultural issues that need to be taken into account. Is probably not this. We should see it clearly from their perspective and not come with just our understanding of how a genetic diagnosis is dealt with. So yes, definitely, this is not easy. We are not participating ourselves in informing the patients, of course. This is up to the clinical team over there. But we know that it has consequences, of course, and we are interested in that impact and how it's dealt in the different countries and different places.
So you're at a point now that you've received this initial tranche of funding from industry groups and the patient support and advocacy organisation, and you have hope to continue and expand this program into the future. At what stage is that sort of advocacy and measuring outcomes and impact and building a business case for additional funding or expansion?
Yeah, that's what we are thinking now, because the other thing that we didn't want to happen with a project like this is that once we're done with the sequencing, then we stop working, and that's all forgotten more or less. That's not the idea. When we thought of this virtual side of the training and the forum and the discussions, was try to build something that will remain, so information, training, knowledge that will remain in those countries. And if at some point they'll have access to additional sequencing analysis, they could maybe work on their own for this. So yes, definitely what we want to do is to the next grants that we want to to apply for will be trying to make the infrastructure, maintain the infrastructure in the future. Something that I think I mentioned briefly is that from my point of view, something that it would be extremely important would be to be able to develop a genetic database where all the genetic data that we collect, but also previous genetic data, can be shared and used by all the centers, because again, we know that there will be variants that will be specific in the region, geographic specific, and that might not be identified initially. That's why I know that I was a bit cheeky when I mentioned that the private companies might not look at the data in the same way. I'm going to stand by my comment. It's just that we came across this issue that variants that are supposed to be of unknown significance are not looked at further. And as soon as you've got a larger population from the same place, you realize that, yes, you do have other patients with variants that are starting to come up again and again. So having a database for all the centers where all this data could be stored would be amazing, and maintaining the infrastructure of the center, of the consortium as such, and the connections would be ideal. But yes, we need to think of how to plan this. Again, we are now trying to convince the centers to apply, for them to apply for money. So it's not seen as this kind of European, white savior project, that it is definitely is not what we are doing, but we have come across people that might see it that way, and that's not the point. We definitely want to help with the patients with diagnosis, and we want to help as much as possible the centers and leave in the on the
from the perspective of sort of variation and those sort of databases, have you engaged with the sort of burgeoning genomic initiatives in the region? Genomics Brazil, and there's activity in Chile, of course.
yes.
Can you leverage their investment in population sequencing initiatives to help resolve some of these variants?
Yeah, that's what we are hoping to do. It's at the moment it's just 3 of us in the project.
Okay.
So we and we are now starting to get all the data through. So yes, we need to see how we manage the workload, and one of us needs to probably just sit down and start writing and contacting our sources for funding and options for the future.
Well, you've achieved extraordinary things with 3 people, just given the scale of work you've undertaken to date. And, we certainly will keep an eye on your program moving forward. It's an excellent initiative, and we really do hope this is infrastructure that helps these countries and these patients and families into the future. So thanks very much for sharing your presentation and project with us, Ana.
Well, thank you very much for having us. Glad you enjoyed it.
Thank you.
Thanks.
And thank you all, audience, for joining us today for this presentation. There will be no DNA Dialogue seminar in July, as next week we're hosting Dr. Eric Green, the Director of the NHGRI, for a special seminar next Tuesday the 2nd at 12 p.m. Eastern Standard Time. So please do, we've certainly been spreading that information far and wide, so please do register and attend to hear Eric. And our next formal DNA Dialogue seminar will be held on Thursday the 29th of August at 9 a.m. Eastern, and will feature Janine Austin from the University of British Columbia, who will be talking about the importance of genetic counselling research and integrating genomics into primary care. So I hope you will join us then. Thank you very much for attending.